Okay, everybody, we're going to talk about cellular respiration today. This is probably a little bit more advanced than you may have had in your introductory biology classes. Just to review some things you might have heard of before, I should have a picture of a mitochondria up. I know you guys are lagging a little bit. I'm watching for that to appear on your screen, and I might be lagged out too much. So I'm going to send you guys a different way to see this. Just hang on. I'm tired of messing with it. We're going to present a window. All right, there you go. You should see that. I'll keep teaching over here for the video side. You guys see a picture of a mitochondria? Presentation mode? Yes, no?
Yeah.
All right, good. So pay attention to that. You just won't be able to see my gestures and face, but that will be in the recording that will come out later. So this mitochondria here, just want you to kind of know that it has an outer area. So it has its own outer membrane here. It has an inner area, which makes up the matrix, but that inner area has deep folds in it to increase surface area, because the membrane of the mitochondria is where all these reactions happen. And the more membrane you have, the more surface area you have, and the more places those reactions can happen. Some other things you might see in here, if you really zoomed in, I'm not sure if your image quality is good enough. There are little rings of DNA inside of the mitochondria. Where else do we see creatures with rings of DNA?
Creatures with rings of DNA?
Yeah, instead of chromosomes. Where else do we see that?
I have no idea.
Well,
Prokaryote?
Yeah, it's prokaryotes. Where do we think that these mitochondria came from? Endosymbiosis, prokaryotes. So this kind of makes sense. More evidence for endosymbiosis. Now, I'm going to go over a big version of cellular respiration. So, cellular respiration is basically taking oxygen in, burning sugar, releasing water and CO2. So you probably know that equation. So cells harvest chemical energy stored in organic molecules. That's the sugar. We generate ATP. ATP is used to do all sorts of cellular processes. Okay? So every organism undergoes some sort of energy generation, but not all of them use cellular respiration all the time. They can do fermentation, they can do glycolysis, but cellular respiration gives a lot more energy. So a lot of people seem to always forget that plants also do cellular respiration. Plants don't just make sugar for us to eat, they make it for them to use. So plants do cellular respiration as well. Okay, so let's stop for a second. Is respiration catabolic or anabolic? Are we tearing molecules apart or putting them together?
Putting them together.
Which molecules are we putting together? Are we going
organic molecules?
Okay, so we're actually breaking the organic molecules down. So that's going to be catabolic. Okay, breaking down. Now we have energy coming out. So is it endergonic or exergonic?
Coming out?
Yeah.
Exergonic?
That's right. So cellular respiration is a catabolic process, and it's exergonic, okay? So keep that in mind. Where do all these organic molecules come from? For most animals, it's starch. It can also be cellulose, but for most of us, it's starch. So sometimes humans will eat refined sugars, which are, oatmeal would be a starch, pasta would be a starch, corn syrup would be more of a sugar. So it just depends on where we're getting things from. Okay, the starch breaks down into glucose, and here's what we have. We have a catabolic breakdown, which I asked you on that last slide, and we worked our way to that. And before we look at this any farther, I want you to just know that these are redox reactions. And in redox reactions, electrons are moving around. And we talked about these two memory tools over here. In oil rig, it is oxidation is lost, reduction is gained. Just make sure you know they're talking about electrons. The second one, a lot of people like better because the E reminds you it's electrons. Leo, loss of electrons is oxidized, and gain of electrons is reduction. So loss and gain. So what we're going to do, we're going to take I think I went backwards. I'm going the wrong way. Okay, what we're going to do is take our sugar and we're going to oxidize it. This is where a lot of people get confused because they think that oxidizing sugar should give you oxygen. Okay? What's happening here is we're oxidizing the sugar, so we're adding oxygen to the carbon. We're adding oxygen to the carbon. The other side, we're going to reduce. So we're going to reduce reduce the oxygen to water. So this is one of those things that students, they see this for the first time and it's confusing. You cannot watch this stuff one time and know it. You're going to probably have to watch this video I'm making, and you're going to probably have to watch some other people's videos, do some practice problems on it, draw some diagrams, and remember, right now you should be taking some notes that you're going to have to turn in as soon as this lecture's over. Okay? So if I'm going too fast, let me know. But
that was on accident, my bad. I just wanted to press it.
You just wanted to see what would happen, huh?
Yeah.
All right.
My bad.
That's okay. So we oxidize sugar, and when you burn something, you can think of like, well, it takes oxygen to burn something. So you can say like, well, I'm burning sugar. What do you get out? You get energy. So the oxidation of glucose, you transfer your electrons. And that step down of electrons, the breakdown, releases energy. And that released energy makes ATP for whatever's, doing cellular respiration. Okay. So during cellular respiration, your electrons are going to flow down, and almost like rolling a ball down a hill or like water going over a waterfall, as they go down, they release energy. And what kind of things do we get? Well, we start with glucose, and I'm going to introduce a new substance here, and you're probably like, well, I saw that NAD stuff. What's missing from the NAD on this equation compared to photosynthesis? If you look back at photosynthesis always had NADPH. And that P, you can remember that because the P, photosynthesis. There's no photosynthesis in cellular respiration, so we're going to take the P out. All right. Let me delete that for now. All right, so we go from the glucose molecule to NADH, which I'm just going to start saying NADH. Then we go to electron transport chain, which we're going to talk about later. This is a big overview. We're going to look at all this again later. And the last step is the electrons flow to oxygen. All right, like I said, your first try at this, you're going to probably be a little confused. It's complex. So when we get this energy, we have multiple steps, and we're going to look at each one in depth. Today, we're just going to look at glycolysis. So we don't just break a glucose down all at once. We do it at different spots, okay? And the first one is taking these electrons with an enzyme called dehydrogenases. They take a couple electrons and protons. Then we're going to do some oxidizing where we take 2 more electrons, okay, and 1 more proton. You're going to transfer that over to NAD and make NADH. And this is another energy storage molecule. You get other protons that are broken off as hydrogen ions, and NADH or NADH is an electron carrier. So it's going to carry these electrons over to the ETC, electron transport chain. There's a lot going on here, but when we break it down into individual steps, it'll be easier. So what's the electron transport chain? That happens in the mitochondria. It's membrane proteins. This is why we have all this folding and all these cristae in the mitochondrial membrane. We need space for these to happen. As electrons move through this system, the electron transport chain, they go through redox. Things are gaining electrons. That's what redox means. As they go through this, they release energy to make ATP. And the final step is to transfer electrons to oxygen. That's the final electron acceptor. It's kind of like the dumpster the electrons end up in, and we get water. This whole thing releases energy. All right. We're going to break this down into 3 stages. Today we're only going to cover glycolysis. So the 3 stages we're going to eventually get to, first is glycolysis. Then you get to step 2, which is pyruvate oxidation. You've probably never heard of pyruvate. Pyruvate is a 3-carbon sugar. It's kind of like that G3P back from photosynthesis. It's a different form though. So that 3-carbon sugar is glucose cut in half. Then you get oxidative phosphorylation. That sounds pretty complex, but we will get to it when we talk about the electron transport chain. So 3 different stages. We're going to break down glycolysis today. The important thing is, where do they happen? What are the reactants? And what are the products? Where do the products go? Okay, so let's start glycolysis here. So glycolysis is where respiration starts. The location is out in the cytosol. So you can kind of see down here, we are outside of the mitochondria. We're not in the mitochondria. We're just in the general cytoplasm of the cell. So what do we have going on? We have, I said this earlier, we're taking glucose, which has 6 carbons, and we're breaking it in half into 2 pyruvates. So pyruvates are a 3-carbon molecule. Pyruvates are going to move on into the mitochondria in the next step. Now, you do not have to know anything at all that's on this slide. But if you look very closely, I'm going to use purple. Up over here, each purple dot is a carbon. So glycolysis is actually all these intermediate steps, okay? Thankfully, none of us are on this biochemistry level where we have to know all this stuff. If you're going to be an actual biologist, especially a cellular biologist or a molecular biologist or biochemist, this is something you're going to have to eventually learn. So if you like all this stuff, great. Okay? At the end, if you look really closely here, there are 3 carbons in this pyruvate. We really only care about going from here to here. We're going to take a shortcut. We just care about what the inputs are and what the outputs are. If you look at that picture carefully, ATP is like the little explosion thingy right here. How many of them are there? It's not a trick question. All right, there are 2 that go in. There's 1 and there's 1. And it says this happens 2 times, so there are 4 that come out. So I want you to see that here. We're going to talk about that. So we don't need to worry about this big picture right here. We're just going to study this right here. We have an ADP that comes in, and for each ADP that goes in, you get 2 ATP out. So this is a good investment. If I said, hey, I have an investment where you can double your money every cycle, you'd probably say, here's my money, double it for me. All right? Ali, what do you got going on?
I accidentally turned on the camera, my bad.
Oh, it's fine. All right, so the other side of this is you've got your electron carrier. This NAD becomes NADH, and you get water out, okay? And at the end of this, you also have your 2 pyruvates, which are your 3-carbon molecules that are going to go on to the next step. So you have some inputs and outputs. Let's look at those, and we'll move on to another. We'll wrap up glycolysis. Okay, so you have an investment stage and you have a payoff stage. So we invest some ATP and we get some ATP out. All right. So for every single molecule of glucose in glycolysis, your payoff is going to be 2 ATPs, 2 NADHs, and also some pyruvate. So take a look here. If you're going to write anything down in this entire lecture, this is what is important. Okay? We're going to invest 2 ATPs. They're going to break down and not be ATP anymore. What are we going to get out? We're going to get out 4 ATPs. So we doubled our ATP. Okay? We put 2 in, we got 4 out. We also get out 2 NADHs, okay, and some hydrogen. So down at the bottom is the net. The net is what happens after all the costs are paid. If you have a paycheck, your net pay is after they take the taxes out. So after glycolysis happens, you get 2 pyruvates, some water, you get 2 ATPs. Now, remember, you had to put 2 in to get 4 out. So that's where the net comes in. And you also get your NADHs. Okay. So I have some practice questions for you. All right. I wrote this so the answers pop up automatically. So I'll just kind of go through these. Where does it happen? That's something I really need you to know is where it happens. It happens not in the mitochondria. If this is a cell, and this is a mitochondria, it's going to happen out here, okay? It's not going to happen in the mitochondria. All right, the next thing. The main things that are produced through glycolysis. First is pyruvate, but what else did I say we get? We get 2 ATPs, and we get NADH, and we get some water. So the next step will be pyruvate oxidation. I'm going to go ahead and end this lecture here. I know a lot of people from the morning class wanted to see a recording of it. You guys might need a recording of it as well. So I'll get this up on the internet here in a little bit.